This is a very special session compared to the others because it is a marathon of fire rapid, very short 4-minute presentations of poster presenters. And we will have to be very, very careful with the time. I will introduce everybody, and then they will share their screen and have their 4 minutes. We don't have time for questions during the session, but we can meet afterwards in the debate lounge number 2, which belongs to the stream number 2, and then we can have an interactive session there. So I think let's start. Before I introduce the first speaker, I just want to warn that Lakshmi Kumar will be the next one, so be prepared for that. Now I would like to introduce the first speaker, which is Laila Arabi from the Mashhad University of Medical Sciences in Iran. And Laila will talk about liposomal celecoxib combined with dendritic cell therapy in B16F10 mouse model for melanoma. The floor is yours, Laila.
Okay. Hi, everyone. Could you see my screen? I've already shared.
Yes.
Okay,
yes.
Thank you very much. Thank you very much for your kind introduction. I'm Dr. Laila Arabi, Assistant Professor of Pharmaceutical Nanotechnology from Iran. Today, I'm going to present the result of combined immunotherapy for treatment of melanoma. As you know, the melanoma is one of the most immunogenic cancers, and it could be considered as a good candidate to evaluate the immunotherapy treatment. Here for this, we try to have combinational therapy to enhance the effectiveness of treatment for melanoma. In one hand, as you see here, let's see my pointer. Yeah, as you see here, the celecoxib would be formulated in liposomes. Why celecoxib? You know that this drug is a non-steroid anti-inflammatory drug and could act as inhibitor of COX-2, which is highly expressed at the tumor site. So the rationale is to inhibit this enzyme, to have the tumor growth regression, and to eliminate inhibitory mechanism of tumor microenvironment. This is one side of the study. The other side of the study is to target antigens to dendritic cells. Again, we used liposomes. Liposomes were decorated with melanoma antigens, GP100 peptide, a bicovalent binding on the surface of liposomes. Then dendritic cells were matured with this liposomal GP100 peptide, and we hope that this peptide could enhance the MHC1 class presentation and also cytotoxic T cell activation. So this is the whole rationale of this study and two sides of combinational therapy. So to test this combination therapy, we performed two models, prophylactic and also therapeutic model in B16F10 bearing C57 black mice. As you see here, for example, for therapeutic model, 10 days after tumor inoculation, mice were injected with IV administration of liposomal celecoxib at one side, and also the injection of the subcutaneous injection of the matured dendritic cells. We repeated 3 times this. We performed this injection for 3 times with 1 week interval. So we performed several immunological tests such as enzyme leak, immunospot assay, flow cytometry, cytotoxicity assay. I just show a couple of them here. All of them on splenocyte suspension. As you see here, for example, this co-administration, this co-administration showed the higher cytotoxicity activity compared to the other group, and also in the therapeutic experiment, as you see here, the combinational therapy, the brown curve here and here, showed effective tumor regression and also enhanced survival compared to the other group. Oops. I don't know what's going on. So altogether, our findings suggest that this combinational therapy could induce immune response, effective immune response, and could enhance the therapeutic outcome, as you could see here in a mouse model of melanoma. And it could be employed as a promising treatment for melanoma. Thanks, everybody, and I'm happy to take any question. I try to just not to be so long. Thank you.
Thank you very much, Laila. And now we continue. Arisiram Ramachandra, please be ready to be the one after that. Now I would like to introduce Lakshmi Kumir from AIMS Pnekarikoki in India. And she will talk about fucoscyanine sorafenib nanoformulation with enhanced oral bioavailability and anti-tumor efficacy against FLT3 ITD acute myeloid leukemia. Please, the floor is yours.
Okay. Welcome, everyone, and thank you, Dr. Smith, for that introduction. So I'll be giving a brief on my work, which is fucoscyanine sorafenib nanoformulation with enhanced oral bioavailability and anti-tumor efficacy against FLT3 ITD acute myeloid leukemia. So as we all know, sorafenib is an oral multikinase inhibitor, which is effective anti-angiogenic as well as anti-proliferative agent and given orally daily. So it's being found that sorafenib is inhibiting FLT3 ITD mutation in case of acute myeloid leukemia. And nowadays, the clinical trials regarding the sorafenib in case of AML is going on forward. But then the issue with sorafenib is like we need to dose sorafenib at a very high dosage on a daily basis because the oral bioavailability of sorafenib is found to be very low. Since the patients are being dosed at a high dose daily, often they are encountered with severe toxicity issues like gastrointestinal toxicity. There are severe hematological and cutaneous toxicities as well. So my work majorly focuses on how to improve the oral bioavailability of sorafenib using a novel protein carrier, which is a fucoscyanine. That's a plant-based protein, and improve its anti-tumor efficacy against FLT3 mutated acute myeloid leukemia. So as you can see in the first slide, the figure A, that's the sorafenib nanoparticle that I have prepared in my lab, which is coming around 217 nanometer. We can see the size distribution is pretty much very tight, and you can see the TEM images of the same, the fucoscyanine sorafenib nanoparticle, as well as the SEM images showing a size of around 200 nanometers. So after preparing this bare fucoscyanine sorafenib nanoparticle, we have coated the particle with a mucoadhesive coating. That is because since the drug is being delivered orally, we have to protect the particle, especially protein-based particle, from the severe gastric harsh pHs. So that's the reason we have given a coating of mucoadhesive coating. And after giving an adhesive coating, you can see the sorafenib nanoformulation. The nanoparticle has got a slight size increase to 329.5 nanometers as seen in figure D. And you can see the TEM images showing a slight coating around the individual particle, giving around a size of around 300 nanometers. And in the SEM images, you can see after the coated particles, there is clusters of small nanoparticles inside the mucoadhesive layer. So since my project basically focuses into the translational development of protein-based sorafenib nanoformulation, which is funded by the government of India, we majorly focus on the batch-to-batch reproducibility, the consistency in the data, and majorly the scalability, how efficient we are, how efficient enough we are to scale up. So the figure G represents the batch-to-batch reproducibility of fucoscyanine phycocyanin sorafenib nanoformulation. Here afterwards, I'll be calling that as a PASORF oral formulation. You can see the batch-to-batch reproducibility in terms of size. As you can see, there is a consistency in the size around 300 nanometers to 300 to 350 nanometers, zeta potential of around minus 50 range, and drug encapsulation efficiency. We have found a very high drug encapsulation efficiency of pretty much around 94%. And the loading efficiency, when I talk, that's with respect to the carrier protein, that's coming around 42%. So that's how we confirm the preparation that we have done has got a very consistent reproducible data. Further, after preparation, we have given this particular oral formulation into Sprague dolly rats orally. To see how the kinetics, the single dose kinetics of this particular formulation look like. So as you can see in figure H, that's the control sorafenib, which is blue line, and the phycocyanin based formulation. You can see there is an enhancement in the formulation. And further, to confirm this, we have gone for a multi-dosing study. Figure H represents a single dose study, and figure I represents a multi-dose study, wherein the dosing was given continuously for 12 days, and blood was drawn in different time points, like 5th day, 6th day, and so on, till 12 days. So our concept of enhanced oral delivery was again confirmed with the multi-dose delivery. As you can see, the blue line is the control sorafenib, and compared to the blue control, our phycocyanin-based sorafenib has already shown.
You have to finish.
Okay, twofold enhancement. And further, we tested in the AML nude mice model. As you can see, there is an enhanced anti-tumor efficacy as well for this particular formulation. And quickly just going through the works happening at my center, these are some of the products that we are getting into the clinical trials. As you can see, the first one is my own product. This is protein-based sorafenib nanomedicine. And these are some of the facilities at Amrita Center for Nanoscience that we have got all the basic R&D facility, characterization, and all the testing facilities for the nano-based testing. And as well as we have got a current nanomedicine GMP facility at ACNSMM, that's state-of-art first GMP-enabled nanotechnology incubator startup cells for the company. Thank you so much.
Thank you very much. Ian Nishunke, please be prepared to be the next one. Now I would like to, if you please could end share so the next one can share, I would like to introduce Arathuram Ramachandra from the University of Birmingham in the UK with a presentation of development of self-powered multifunctional piezomagnetic nanoparticles for non-invasive BBB modulation and glioblastoma treatment. Please, the floor is yours. You are muted. We can see your slides.
Yeah,
but,
yeah. Sorry. Thank you, Schmid, for the kind introduction, and welcome all for a short discussion about my work. So I'm Arathuram, as she mentioned. I'm working as a medical fellow at the School of Pharmacy, University of Birmingham. So as you know, blood-brain barrier protects the brain from unwanted chemicals, but it often presents a barrier, tight barrier to cross the therapeutic molecules into the brain for the treatment of several neurological disorders, as well as glioblastoma, especially glioblastoma and other brain tumors. So I am proposing to develop an ultrasound sensitive nanocarrier system for the treatment of glioblastoma especially. So here I am introducing the nanoparticle called piezomagnetic nanoparticle system in this. The piezomagnetic nanoparticles are containing the piezoelectric core and the superparamantic iron oxide nanoparticles as a shell to give the extra magnetic chargeability as well as the non-invasive ultrasound penetrability. So the main idea is when I modulate this nanoparticle with ultrasound, non-invasive ultrasound, this nanoparticle can produce electrical signals. These electrical signals can help to penetrate the tight junctions in the endothelial cells of the blood-brain barrier. That is my idea. So these are some of the characterizations, and we found the nanoparticles are very effective in producing electrical signals when it is stimulated with ultrasound. And as a proof of concept for the glioblastoma treatment, we conjugated an anti-cancer drug, cisplatin, along with this piezo-magnetic nanoparticles, and this is the bonding is confirmed by the FTIR spectroscopy. So as I mentioned, for the BBB penetration, we prepared because we know that the organoids and tumor spheroids are gaining wide attention for the drug discovery treatments. So we developed a blood-brain barrier spheroid using the main cells from the neurovascular unit. So that are the astrocytes, pericytes, and the endothelial cells. As we hypothesized, these systems comprise same in cellular organization as of the blood-brain barrier, with astrocytes in the middle. As you can see here, the red color is astrocyte cells, and it is covered by encasing the surface with the endothelial cells and the pericytes with a tight junction. So it can perform the same regulation property. Transport molecules are confirmed. So the tight junctions also confirm the high organization of the blood-brain barrier. So now we tested the efficacy of our nanoparticles to penetrate deep inside the blood-brain barrier. As you can see here, when it is stimulated with ultrasound, the nanoparticles, this cyan color is from the nanoparticles, has the own second harmonic generation imaging property. So the nanoparticles are visualized by its inherent second harmonic generation imaging, and we found that the ultrasound treated samples are very effective in penetrating the blood-brain barrier. And also we tested its efficacy to penetrate deep inside the glioblastoma spheroids, and we found that the ultrasound treated nanoparticles are going deep inside the spheroids for the effective treatment of treatment of tumor using the drug delivery system. So we tested the efficacy of our nanocomposites for the treatment of glioblastoma using cell-culture glow 3D, and we found that the nanoparticles stimulated with ultrasound and showed enhanced anti-cancer effects compared to the untreated ultrasound controls. And the bright field images and the live images are clearly confirmed what we have expected. So that means the enhanced penetration of the nanocomposites due to ultrasound, as well as the ultrasound-induced anti-cancer delivery resulted in the enhanced therapeutic capability. So in short, I'm going to conclude that these nanoparticles are very effective as a future candidate for the treatment of various disorders, not only for the brain-related disorders, those tumors which are hard to reach. We can use this nanoparticle with non-invasive ultrasound and the nanoparticle. Thank you very much.
Thank you very much. Valeria Sidorenko, you can start preparing. Now I would like to introduce Jenny Schunke from the University Medical Center of the Johannes Gutenberg University of Mainz in Germany, and she will talk about multi-component adjuvantation of antigen-based nanocapsules using site-directed click chemistry cross-linking for the treatment of melanoma.
Hello, everyone. I hope you can see my presentation. And
we see the slide mode where you see the next slide. We don't see the.
Okay, I share my screen again. Now it's correct.
Yes, now it's good.
Okay.
Thank you
for the kind introduction. I'm Jenny Schunke, a PhD candidate of the University Medical Center in the group of Volker Mailänder. And today I would like to present you my work, which is about the antigen nanocapsule-based combinatorial delivery of the STING and Toll-like receptor 7 and 8 agonists, which enhances anti-tumor immune responses. First of all, it's still important to develop new treatment options for cancer treatment, because many of those treatments which are available are limited or show strong side effects. So this is why we are working on vaccination approaches based on the targeting of dendritic cells, which are the most important antigen-presenting cells with overwhelming nanocarriers, and they enable the combined transport of antigen and adjuvants. And in my case, I'm working with the APTI, a sting agonist, and Resiquimod R848, which is a toll-like receptor 7 and 8 agonist. In vitro, we treated bone marrow-derived dendritic cells with different nanocarriers and measured their uptake, the dendritic cell maturation, also the secretion of pro-inflammatory cytokines. And we could show a strong uptake and increased expression of so-called maturation markers like CD86 and CD80 after the treatment with the APTI and the double combination loaded nanocarriers by flow cytometry, as well as high amounts of different pro-inflammatory cytokines by combining the APTI and R848 in one ovalbumin nanocarrier, which you can see on the left side in black. So we further tested our DAPT and R848 loaded ovalbumin nanocarrier in an B60 melanoma model, and therefore we injected ovalbumin expressing B16 cells subcutaneously into the mice flanks. And we then treated the tumors 3 times when they had a size between 25 or 50 cubic millimeters. And we could show that the mice which received the treatment with the APTI and R848 loaded nanocarriers were cured and showed a prolonged overall survival. Our results showed additionally the advantage of encapsulating the adjuvants compared to injecting soluble adjuvants. But unfortunately, we just could re-challenge 2 mice with 100,000 cells after 90 days because the others developed the necrosis of the back skin, but we already fixed the side effects by reducing the injected dose. But the tumors did not grow after the re-challenge, which shows that we have an effective immune memory. In another experiment, we observed that the tumor treatment with DAPT and the double combination loaded nanocarriers increased the amount of tumor infiltrating monocytes as well as cytotoxic T lymphocytes by simultaneous reduction of PD-1 expression on CD8 positive T cells. And the amount of pro-inflammatory cytokines like IF and gamma was also strongly enhanced in vivo. So we would furthermore like to test the nanocarrier application in an over-independent melanoma model by encapsulating tumor-specific antigens. And we want to test the antibody-based immune cell targeting with those capsules and of course optimize the vaccination scheme and also encapsulate other adjuvant combinations. Thank you for listening, and yeah, I would be happy to take some questions afterwards.
Thank you very much. Katarina Spiridopoulou, you have to start preparing. And now I would like to introduce Valeria Sidorenko from the University of Tartu in Estonia. She will talk about tumor-penetrating uterobicin polymersomes for cancer therapy.
Yes. Hello. Thank you for the nice introduction. And here's the overview of my poster, which is about, as I said before, is about tumor penetrating and the uterobicin loaded polymersomes for cancer therapy and detection. Okay, are the slides moving? Yeah. So uterobicin, or shortly the UTO, is a novel fluorescent and hydrophobic anthracycline developed by Estonian company TBD Biodiscovery. UTO is more potent than clinically used doxorubicin in a panel of cultured cancer cells because of its modified molecular structure. So it has the anthraquinone backbone that intercalates the cell DNA and also can induce the formation of reactive oxygen species inside the cancer cell. In addition, it has a reactive oxazolidine cycle, which after the cleavage of the enzymatically cleavable protecting group, covalently binds to the DNA base guanine. And to enhance the potency of UTO, the hydroxyl group is added, making it a more potent metabolite in vivo, and the amino group is added as it is found to be less cardiotoxic. So the UTO and its nanoformulation are already patented and published in Angewandte Chemie. And to improve the biodistribution and tumor homing of UTO, we encapsulated it into polymeric nanovesicles or polymersomes, which are composed of biocompatible, biodegradable, and also FDA-approved polymer called polyethylene glycol polycarbolactone, or simply PEG-PCL. And after, we functionalized these polymersomes with a CN rule, or shortly sender peptide, which gives us the peptide targeted and auto-encapsulated polymersomes. So sender peptides act as tumor-penetrating peptides that home and bind to the neuropilin-1 receptor, which is overexpressed on the surface of many cancer cells and also tumor blood vessels. And such binding triggers then the penetration of the peptide along with the cargo into the tumor tissue. And we found that in cultured sender receptor positive cancer cells, our peptide targeted and UTO loaded polymersomes killed significantly more cancer cells than non-targeted UTO particles, showing that the anti-cancer effect was potentiated by the binding of the sender peptide to the neuropilin-1 receptor. And to assess the anti-cancer effect of nanoformulated UTO, we performed an experimental treatment in mice bearing peritoneal carcinomatosis of gastric carcinoma origin. So mice were intraperitoneally injected with UTO-loaded polymersomes every other day, starting from the day 4 after tumor inoculation, and the total dose of UTO was 13 milligrams per kilo. And we found that UTO polymersomes significantly suppressed the tumor growth without showing signs of systemic toxicity, as the body weight did not differ from the control group treated with PBS. And encouraged by these results, we are currently evaluating the therapeutic activity of the peptide-targeted and UTO-loaded polymersomes, and we expect to achieve even more potentiated anti-cancer effect. And I want to thank our group from the Laboratory of Precision and Nanomedicine, also our sponsors, and all of you for listening, and thank you.
Thank you very much.
Mm-hmm.
Now, Shiki Wang, you can start preparing. And now I would like to introduce Katarina Spiridopoulou from the Democritus University of Thrace in Greece. She will talk about diverse bioactivities of biogenic SENPs spur cancer cell-based vaccine potential.
Hello, everyone. Can you hear me?
Yes.
Okay, great.
It's not in presenter mode. Can you put it in presenter mode? Otherwise, it's readable if it's not possible.
It is. Don't you see the whole? It is in presenter mode actually.
Not.
It's not.
Not for us. No.
Oh, let me do it one more time. Okay. How about now? Is that okay?
We can see it. Just continue that way. It's not in presenter mode, but it's big enough, I think, to see. Thank you.
Okay. Okay. I'm sorry for this. Just a moment. Okay. So excuse me for the delay. Thank you for the kind introduction. Really happy to be here today. So I will talk about the diverse bioactivities of biogenic selenium nanoparticles that spur cancer cell-based vaccine potential. So why selenium? Selenium exerts multiple health-promoting effects, among which are anti-cancer activities and also immunomodulatory activities. But unfortunately, in high doses, it also exerts toxic side effects. So a more bioavailable and more safe alternative is selenium nanoparticles. So we achieve the biogenic synthesis of selenium nanoparticles by blowing the probiotic strain Lactobacillus casei ATCC393 And we chose this specific strain because based on our previous work, we had seen that the strain itself exerts anti-tumor effects against colon cancer, and the bacteria also can accumulate selenium. So
sorry, we still see the first slide.
I'm so sorry.
Now we see the second. Now we see the second one.
Okay, maybe I'm not in presenter mode though, so maybe I should just go on this like this, leave it in this mode, and go on from this. What do you think?
Yeah, but probably you are on the third slide now when you talk. I'm not sure.
Yes. Why? Let me try something else. I'm so sorry for the delay. How about now?
We see the second slide about selenium. But if you just move to the next slide.
Okay, here. Do you see the whole slide? So the outline here.
We don't. No.
Okay, maybe is that okay if you go on to the next presenter and I'll come up next? So in order to fix this, I don't know why this is happening. I'm really sorry.
Yeah, we can do that. So in that case, you have to unshare, and in that case, I would like to introduce Shiki Wang from the University of Helsinki in Finland, and she will talk about polyoxometallate incorporating injectable hydrogel with pH and NIR responsiveness for hemophotothermal therapy.
Thank you for the kind introduction. My name is Shiki Wang, and I'm from the Faculty of Pharmacy, University of Helsinki. Today, I'm going to present my recent work about this polyoxometallate incorporating injectable hydrogel for chemophotothermal therapy. So first, let me introduce about the project design. So what is polyoxometallate? It is kind of a small nanocluster made of metal and also some other elements. And here, more specifically, I'm using Mo156. So as you can see here, it is a very small almost 4 nanometer metal cluster, and it has a very advantageous property, which is the photothermal capability, meaning if we shine a near infrared on this Mo156, the temperature will significantly increase compared with the other like commonly used photothermal reagents like gold nanoparticles. Mo156 has much more higher like photothermal capacity. However, there is a big issue of using this Mo156 in the biomedical applications, mainly about the stability of the molecule and also the toxicity. So in this project, I'm incorporating Mo156 in an injectable hydrogel. So I'm using two biocompatible polymers in their modified version. One is PEG with aldehyde groups in the end, and also the other one is chitosan. So as you can see here, the aldehyde group from the PEG and the amine group from the chitosan can form this dynamic shift base, non-covalent cross-linking. And also we can introduce some drugs into the hydrogel to make it a drug delivery platform. So how this project is related to nanomedicine and targeted delivery. So first of all, this hydrogel preparation is very simple and fast. So at room temperature, by simply mixing all the components, we can have the gel in less than 1 minute. And as you can see in the picture, the gel is injectable through this syringe and needles, and the whole process is very easy. And also, or more importantly, by incorporating in the gel, we managed to improve the stability and biosafety of Mo156. So as you can see here in the drug release study, we can apply multiple time of NIR, which is here 808 nanometer wavelength irradiation, and then the drug release can be controlled, and then the MO156 still have photothermal capacity after several cycles of laser irradiation. And we also tested the toxicity on primary human fibroblasts, and with the gel encapsulation, the biocompatibility significantly improved. And finally, we applied the self-healing bioinjectable biodegradable hydrogel in an in vivo proof of concept study for chemo and photothermal therapy. So as you can see here in a melanoma mice model, we managed to achieve quite significant tumor suppressive effects in the end by combining the chemo and photothermal therapy of this gel, and the body weight doesn't change much. And I'm happy to share with you all more data, and if you have any question, I'm happy to discuss in the end. Thank you.
Thank you very much. Katarina, are you ready now, or should we take one more other? Anyway, Ahmad Balol, Ahmad Balol has to start getting ready. So then, Katarina, it's your floor again.
Okay, hopefully it works now.
It does. It does now. Yeah.
So, does it work?
Yeah, I think so.
Are the slides changing? Okay.
It's now the second slide.
Okay, so let's pick it up where I left it. So I left it here. I was talking about I was about to describe the outline of our work. So I was saying that we synthesized the selenium nanoparticles inside the probiotic bacteria. We extracted them and characterized them, and investigated their anti-cancer properties in colon cancer cells. And also we investigated the
I think you have to go to the next slide, because now it only says background.
Okay, I'm so sorry. I don't know what's wrong. Maybe you can go on, and I will be the last one, the last presenter, if that is possible, so I can see what's really happening.
Okay.
Okay.
Yeah, so, I'm sorry.
No, that's okay. That's okay. Please go on.
Yeah. Okay. In that case, Carolina Medina Montana can prepare to get ready. And now I would like to introduce Ahmad Balol from the Royal College of Surgeons in Ireland. He will talk about development of inhalable retinoic acid-loaded polymeric nanoparticles as targeted host-directed immunotherapy for Mycobacterium tuberculosis. Please.
Thank you for the kind introduction. Hello, everybody. And thanks for giving me the opportunity to give an overview about my PhD project developing inhaled host-directed therapy for tuberculosis. I'm a PhD student at the Royal College of Surgeons in Ireland and St. James's Hospital. So TB is the top bacterial infectious killer disease in the world, with 1.5 million deaths in 2020. The current COVID-19 pandemic is significantly impacting TB case finding and access to treatment, which would worsen this number. The anti-TB drugs have serious side effects, which leads to poor patient adherence and the rise of multi-drug resistant and extensive drug-resistant TB cases. So there is an unmet clinical need to develop new treatment strategies to overcome the issue of multi-drug resistant TB. One of these strategies is to develop host-directed therapies, which acts on boosting the host immune system rather than acting on the bacteria itself. One of the emerging host-directed therapies from our lab is all-trans-retinoic acid, or ATRA, which is the active metabolite of vitamin A. It's currently FDA approved for treatment of acne and acute promyelocytic leukemia. So we are repurposing this drug for TB. ATRA works on TB by several mechanisms: induction of autophagy, depletion of the bacterial intracellular nutrients, modulation of the cytokine profile and signaling, and augmentation of immune cells. However, this molecule is lipophilic, so it needs a formulation step before it can be in vivo administered. So the aim of my project is to encapsulate ATRA into PLJ nanoparticles and to target it via inhalation to the alveolar macrophages. We formulated our nanoparticles with a size of 250 nanometers and almost 70% encapsulation efficiency, and we were able to scale it up and optimize it using the Ignite nanoassembler microfluidic system for scaling up. And then we moved on to assess the efficacy in an in vitro infection model, and we found that our nanoparticles were able to decrease the bacterial growth in vitro in a dose-dependent manner. Then we moved on to assess the safety of our nanoparticles. So we've assessed the cell viability on both macrophages and alveolar epithelial cells. And our nanoparticles showed no toxicity on both airway cells. Then we moved on to integrate our nanoparticles with our industrial collaborator with a vibrating mesh nebulizer in order to deliver it by inhalation. And we've assessed the nebulized nanoparticle droplet size, and our nebulized nanoparticles showed an optimum droplet size required for lung deposition with a volume median diameter of 4 micrometer, as shown by Spraytech data by laser diffraction. And we've also characterized the aerosol using cascade impaction, and we found that almost half of our delivered dose deposited in the regions of the impactor representing the terminal bronchi and in the alveolar region with a mass median aerodynamic diameter of 2 micrometer. Then we moved on for further translational purposes to assess the inhaled dose in a breathing simulator experiment. So we've connected our nebulizer to a breathing simulator, and we've generated an adult breathing profile pattern to assess the inhaled dose. And we found that 65% of our dose were inhaled, which is higher than many commercially available inhalers. So we're thinking that those nanoparticles could be targeted with high efficiency, with high delivered dose to the lungs. We are planning as a future perspective to move on in vivo in a clinically relevant in vivo mouse model with our collaborators at Imperial College London. So if you are interested in our project, please pass by my poster at page 5 on the poster session, and pass by also our recently published review, All Transcitanoic Acid as Host-Directed Immunotherapy for Tuberculosis. And I'm happy to chat with you about my project in the debate lounge later. And thank you so much.
Thank you very much. Yanira Sain, you can start getting prepared. And now I would like to introduce Carolina Medina Montana from the University Medical Center of the Johannes Gutenberg University Mainz in Germany. She will talk about biological effects of systemically administered TLR7/8 agonist and antigen conjugated nanogels on the immune cell populations in the liver and in the spleen. Please, the floor is yours.
Hello, everybody. Can you hear me?
Yes.
Perfect. Good morning. My name is Carolina Medina Montano, and I'm a PhD student at the Department of Dermatology at the University Medical Center in Mainz. And today I want to present you a part of my work regarding to the biological effects of nanogels functionalized with toll-like receptor 7/8 agonist and an antigen of immune cell populations. Here we introduce a pH-responsive nanogel platform that allows the attachment of an antigen, in this case ovalbumin, and an adjuvant IMDQ into the same nanocarrier. First, we performed some in vitro experiments. As you can see, we could confirm uptake of the different nanocarriers into the different immune cell population at different extent. We also analyze maturation markers expression of immune cell population, especially of MCD86 and MHC2. And so you can see all IMDQ containing nanoparticle formulation increase the expression levels in dendritic cells, B cells, neutrophils, and macrophages. Interestingly, none of these markers could be stimulated in liver immune cells. Based off these results, we assess biodistributions of the different nanogel formulations, and we observe that the majority of the particles accumulated in the liver and in the kidneys. Ex vivo, we monitor, Ex vivo, we monitor the liver enzyme levels to evaluate potential toxicity after intravenous injection, but we could observe any significant alterations. Moreover, histopathological analysis of hematoxylin eosin staining, which we could show that there is no histological anomalies after intravenous injection. We also monitor the cytokine profiles, and we observe that the nanogels functionalized with IMDQ induce the highest levels of pro-inflammatory cytokines like TNF-alpha, interferon gamma, and IL-6. All of these experiments were carried out within the framework of the SFP 1066 in collaboration with the Department of Dermatology and the MPI here in Mainz, with the objective to develop nanoparticles for the immunotherapy of malignant melanoma. And with this goal, chemists and biologists, we are working together in these complementary and closely areas. And that's all for today. I hope you could enjoy my talk, and thanks for listening.
Thank you very much. Michael Fichter, you can start getting ready. But now I would like to introduce Yanira Zain from the University Medical Center of the Johannes Gutenberg University. Again, we go to Mainz, and she will talk about comparative analysis of nucleic acid-based adjuvants for the activation of dendritic cells to improve nanovaccines.
Oh, 1 second. Sorry. So I hope you can see my presentation now.
Yes, now okay. It's okay. Perfect, thank you.
Okay, then thank you very much for the kind introduction. My name is Yanira Zain. I'm a PhD student at the Department of Dermatology here in the University Medical Center in Mainz. And today I'm going to talk about the start of my PhD project, comparative analysis of nucleic acid-based adjuvants for the activation of dendritic cells to improve nanovaccines. So liver non-parenchymal cells, also short NPC, comprising Kupfer cells, DC, and the specialized endothelial cell subpopulation termed liver sinusoidal endothelial cells, or short LSEC, exert tolerogenic activity by default. All liver NPC express a variety of pattern recognition receptors, short PRR, including those that are expressed by DC in the secondary lymphoid organs. These receptors are often used as targeting modalities for DC addressing nanovaccines, for example, C-type lectin receptors like CD206 or CD209. Among other factors, the overlapping expression pattern of PRR may be one of the major reasons for the unintended predominant accumulation of nanovaccines in the liver. It's conceivable that nanovaccines, on the one hand, may induce adaptive immune responses in the lymph nodes and spleen by targeting dendritic cells, but on the other hand, may cause antigen-specific tolerance upon the engagement of liver NPC, limiting the extent of anti-tumor T-cell responses. Therefore, our project, besides targeting and activating DC in the secondary lymphoid organs, aims to actively reprogram liver NPC towards a T effector cell inducing immunophenotype by applying adequate adjuvants and combination of adjuvants. So in the first phase of the project, we monitor the efficacy of nucleic acid-based adjuvants, here CpG oligos, with regard to the activation of FLT3 ligand differentiated DC and also GM-CSF differentiated DC. Therefore, we monitored the expression of the co-stimulatory receptor CD86, and the DCs were incubated with different doses of the respective CpG oligos. And what we found was that we see a DC population-specific stimulatory efficacy in a market-dependent manner. And also we had a look at cytokine levels of the dendritic cells. And we found that CpG oligos induce pro-inflammatory cytokine release by dendritic cells. For example, if you have a look at interferon alpha. So to this end, we want to employ, on the one hand, already evaluated liposomal formulations obtained from our Department of Chemistry and also from BioNTech, which will contain antigen encoding mRNA plus nucleic acid-based adjuvants. The targeting of the liver NPC and DC in secondary lymphoid organs will be achieved either due to the intrinsic properties of the liposomal formulations, for example, size and zeta potential, or via trimethylisation targeting moiety. Yes, we will get those particles from the Department of Chemistry. And then in accompanying functional assays, the T cell stimulatory capacity of DC and liver NPC will be tested in OT1 and OT2 cells. And we will assess then the therapeutic suitability of the in vitro optimized nanovaccines in a melanoma mouse model bearing clinically relevant mutations. Topical inoculation of the cauding melanoma cells will result then here in melanoma, whereas the injection will then result in liver metastasis. So thank you for listening.
Thank you very much. Hanuman Geikwat, you can start getting ready. And now we still stay at the Medical Center of the Johannes Gutenberg University in Mainz. And now it is Michael Fichter that will talk to us. I think, Janira, you have to unshare. Okay, so now Michael Fichter will talk about achieving dendritic cell subset-specific targeting in vivo by site-directed conjugation of targeting antibodies to nanocarriers. Please, the floor is yours.
Welcome, everyone. Can you hear me?
Yes.
Nice. My name is Michael Fichter. I'm a postdoc in Volker Mailander's group at the University Medical Center in Mainz at the Department of Dermatology. And in the following minutes, you will get an overview of our project focusing on the optimization of antibody-based targeted delivery of nanocarriers to dendritic cells. Dendritic cells are the most important cells for the induction of antigen-specific immune responses. Therefore, a targeted delivery of agents like antigens or adjuvants to diseased dendritic cells is the main focus of various research projects. While delivery to the other cell types can lead to unwanted effects in the above-mentioned settings. Conventional conjugation techniques, however, result in an undirected and unoriented coupling of the antibodies onto the nanoparticle surface. And this can be a problem since a lot of cell types express FC receptors. This class of receptors can non-specifically bind FC domains of antibodies. And for example, macrophages or Kupffer cells in the liver do express FC receptors at a high level and can bind the nanoparticle. And this can lead in the end to an internalization by non-targeted cells. Therefore, a site-specific and oriented conjugation of antibodies onto the nanoparticle surface is needed to ensure uptake by targeted dendritic cells. And in this project, we established a method for site-specific conjugation using magnetic nanoparticles with a hydroxyethyl starch shell and primary amines on the surface. In the first reaction, a DPCO PEG4 NHS linker was attached. And in a second step, we made use of the carbohydrate groups that are only present at the FC domain of antibodies. And using two enzymatic reactions, we attached A-site groups to each carbohydrate group of the antibody. And finally, copper-free click chemistry was used to stably conjugate the antibodies to the nanoparticle surface. And the targeting capability of antibody functionalized nanocarriers was tested using first the pan-DC antibody anti-CD11C, as well as DC subtype specific antibodies against XCR1, CLEC9A, and DEC205, in addition to a non-specific IgG that was used as a negative control. Nanocarrier formulations were injected intravenously into mice, and uptake by dendritic cell subtypes was evaluated by flow cytometry. Anti-CD11C functionalized nanocarriers were taken up to a high extent by CD11C positive dendritic cells. And as a control for specificity, macrophages that are negative for all the targeted molecules were analyzed as well. And none of the applied nanocarrier formulations exhibited an increased uptake by these cells. Anti-GLEC9A functionalization efficiently targeted conventional dendritic cells type 1, as well as plasmacytoid dendritic cells in the spleen. For conventional disease type 2, anti-CD11C targeting led to the highest uptake rate, while DEC205 showed a slight increase in uptake. Unfortunately, anti-XCR1 functionalization did not provoke an improved uptake by CD11C, by conventional disease type 1 as intended. In summary, here we developed a site-specific antibody conjugation technique for the functionalization of nanocarriers, and anti-GLEC9A conjugation exhibited an efficient targeting of conventional disease type 1 and plasmacytoid dendritic cells, while XCR1 and DEC205 did not prove to be suitable targets using this system. In future studies, we would like to increase the targeting specificity by using nanobodies and combinations of antibodies. Also, we are translating this conjugation technique to more relevant nanocarriers, like polymeric nanocapsules, for example, that also carry antigens and adjuvants for vaccination strategies and the generation of robust anti-tumor immune responses. Yeah, thank you for your interest in this project, and feel free to contact me afterwards for any questions or discussions. And I also thank all the people involved in this project. And yeah, thank you very much.
Thank you very much. Giovanni Cetani, you can start getting prepared. But now I would like to introduce Hanuman Geikwat from the University of Colorado in the US. And he will talk about lipid nanoparticle formulation of niclosamide. Nano, NCM, effectively inhibits SARS-CoV-2 replication in vitro. The floor is yours.
Hi. Yeah, thank you for introduction. I'm going to present my poster on the lipid nanoparticles formulations of nucleosamide. It's called as a nano NCM that can effectively inhibit the SARS-CoV-2 replication in vitro. The nucleosamide is an old anthelmic drug with pleiotropic pharmacological activities. And it's a nucleosamide, it's emerging as another potential repurposed drug candidate to treat the COVID-19 disease. The nucleosamide is a completely water insoluble and which limits its clinical use because the nucleosamide is the class 2 type of drug and it has a poor bioavailability. The limited, it's due to poor biology, it's limited its use. Our idea is to develop cost-effective lipid nanoparticle formulations of nucleosamide by using the FDA-approved excipient and potentiate against the SARS-CoV-2 infected cells. This is the structure of nucleosamide, and this is commercially available the nucleosamide pills. We prepared the nano formulations of the nucleosamide by using the FDA-approved DSP 2000 at a concentration of 10 millimolar concentration and in the basic ethanol to give a fully clear dispersion at 11.1 milligrams per ml and the 3.3 millimolar concentrations of DSP 2000. Further, we subsequent dilutions in the next step with the tenfold dilution step. Due to water, the formulation is presented as clear and colloidally stable dispersion. The final step is done with immediately after the dilutions. The dispersion was neutralized with equimolar HCl to clear the formulations. And we can see the efficiency of formulations around 1.19 to 1. So very high concentrations of nucleosamide can be encapsulated. And the images we can see at 3 different stages the formulations. And further, we studied the stability of this nano-NCM formulations, and which says that it's stable for the 400 hours at 4 degrees centigrade and the 200 hours at the room temperature, but it is slightly less stable at room temperature. Then we checked the encapsulation efficiency of lipid nanoparticle by ultracentrifugation, and it shows that most of the nanoparticles are encapsulated. Most of the nucleosides are encapsulated. And further, we checked the encapsulation efficiency and selectivity towards the SARS-CoV-2 virus. We used the E6 cells, which is derived from monkey kidney and are commonly used for the testing of antiviral agent in the virus-infected cells. The nano-NCM formulation demonstrated the activity with around 0.59 nanomolar acidity values and 3.3 micromolar IC90 values, with achieving the selectivity. And finally, in summary, we developed a simple process for the water-soluble nanoformulations of nucleosamide in 5-minute process using lipid nanoparticles with stability, with a higher efficacy and selectivity for the SARS-CoV-2 inhibitions. The resulting formulations can be scaled up and tested for the preclinical models in the COVID-19 patients, and due to its lack of solubility, it will benefit the formulations. And for more details, you can find in the posters. And thank you for your attention.
Thank you very much. Piotr Tarak can start getting ready. And now we go back to Mainz, but this time to the Johannes Gutenberg University. And I would like to introduce Giovanni Cetani, who will talk about pH-dependent behavior of ionizable cationic lipids in mRNA-carrying lipoplexes investigated by molecular dynamic simulations. The floor is yours.
Can you hear me? Can you see my slides?
Yes. Everything is perfect. Thank you.
Okay, so let me first acknowledge my collaborators, the students working with me and my boss, Friederike, and also my collaborators at BioNTech. So the idea here is the following. We know that it's possible to form lipid-based nanoparticles, or in some cases also polyplexes, by mixing in a well-defined setting messenger RNA and lipids. And then these nanoparticles are supposed to enter the cell, release the messenger RNA, and let the ribosome translate the proteins that we are interested in. The problem is that any information that we get out from experiments is always or mostly rather indirect, both on the structure of the lipoplexes, the lipid-based nanoparticles, and also on the process by which these nanoparticles enter the cell. So the idea here is to use computer simulation to shed a bit of light in this direction. So we use big computers to simulate the lipoplexes or lipid-based nanoparticles, and more recently also the processes involved in transfection. Today I will talk about the internal structure of lipoplexes. So experimentally, it is known that they form lamellar structures. So these lipoplexes have two phases, one rich in lipids and one rich in messenger RNA, separated by a well-defined distance that depends on pH. Now, what we did in our simulation was to reproduce in the computer this structure, this lamellar structure that is observed experimentally, and try and see how, at the molecular level, lipids interact with messenger RNA. The lipids that we are talking about in this case are DODMA, which is a ionizable cationic lipid, and the OPC. So the result that we obtain in this case is the following. At low pH, when most of the DODMA ionizable lipids are charged, these lipids tend to accumulate around the messenger RNA. In this picture, this is yellow. And they form an effective attractive interaction. So there is an effective attractive interaction between ionizable lipids and RNA at low pH. But as soon as the pH increases, these lipids partly change their charge. So the ionizable lipid become less and less charged. And eventually at high pH, they lose completely the charge. And under these conditions, RNA is not anymore effectively attracted by these lipids, and actually it is repelled by these lipids. So we measure an effective repulsion between RNA and cationic lipids, ionizable cationic lipids in their unprotonated state. So the attractive interaction is only present at the low pH. From this calculation, we also observe another interesting thing, that is how RNA changes shapes and position inside the lamellar phase during this process. So at the low pH, when it is attracted by the ionizable lipids, RNA sticks on the lipid bilayer, while at high pH, it basically is found mostly in the water phase, separating the bilayers in the lipoplex. From the point of view of the cationic lipid, the cationic lipid at high pH becomes uncharged, and it can much more easily cross the leaflets. So you can flip from one leaflet to the other. And now what we are going to do is to test many other ionizable lipids in the same setting to understand how the shape of the head group of the cationic lipid and the tail group change the way messenger RNA and the cationic lipid interact, and how this is related to the transfection efficiency that is then measured experimentally by our collaborators at BioNTech.
You have to finish now.
Sorry?
You have to finish. Your time is up.
Yes,
so...
I'm finished.
Okay. Okay, then thank you very much.
You're welcome.
Alice Spadea, could you please start getting ready? Now I would like to introduce Piotr Tarak from the University of Łódź in Poland. He will talk about application of lysine-based peptide dendrimers D3K2, D3R2, and D3H2 for gene delivery, a functional transfection study in vitro.
Hello, everyone. Thank you for the introduction. I would like to present the topic of my poster called Application of Lysine-based peptide dendrimers for gene delivery, functional transfection study in vitro. I'm Piotr Tarach, PhD student in the Department of General Biophysics of University of Łódź, and this work was made under the supervision of Dr. Anna Janaszewska. Our work was done in collaboration with the University of Tajikistan. The peptide dendrimers were synthesized by the research team led by Professor Igor Nilov. And my research material is polylysine dendrimers, which are linear peptide composed of consecutive amino acids attached to the multifunctional organic core. And these amino acids are lysine in the case of D3K2, arginine in the case of D3R2, and histidine in the case of D3H2. And the workflow of the scientific process was as follows. In the first step, the in silico molecular dynamic simulation of lysine-based peptide dendrimers was made. In the next step, biophysical characterization of polylysin dendrimers were made by NMR studies, zeta potential measurements, and hydrodynamic diameter measurements. In the next step, preliminary in vitro studies were made following the cell viability, cellular uptake, and DNA damage tests. In the following step, we performed preliminary studies on in vitro transfection with fluorescently labeled material, followed by flow cytometry and confocal microscopy studies. And in the final step, that's what I'd like to present today, we performed functional in vitro transfection of siRNA targeted against NF-kappa B, and this was measured by real-time PCR and Western blot. We showed that polylysin peptide dendrimers can silence the expression of NF-kappa B subunit called P65 when used to transfect cells with siRNA targeted against this NF-kappa B subunit. Polyalpha-lysin transfected cells with the anti-P65 siRNA had reduced expression of P65 subunit comparable to the lipofectamine 2000. So we complexed the siRNA with the following dendrimers, and we observed the reduction of the expression of the targeted gene. And these results suggest that dendrimers may have the potential for the future development of gene therapies, or may be used as a replacement for lipofectamine 2000. I would like to say a few words about outlook toward translation. So we plan to extend our in vitro studies toward the cell lines, normal or cancerous, for example, Cacao cell lines, as a target for example, for use for oligonucleotides to inhibit, for example, cancer cell invasion of Cacao cell line. For example, to complex our dendrimers with anti-mRNA 21. And in the final step, we would like to perform in vivo animal studies to check the toxicological profile, biodistribution, pharmacokinetic, and pharmacodynamic profile. Thank you for your attention.
Thank you very much. Aldo Namzuk, start getting ready. Now I would like to introduce Alice Padilla from the University of Manchester in the UK, and she will talk about novel insights on endosomal escape of lipid nanoparticles using reflectometry techniques. The floor is yours.
Thank you for the introduction, and hello everyone. So lipid nanoparticles are considered one of the most successful carriers developed for RNA delivery, especially after their success as COVID-19 vaccines. To be effective, the carrier system must protect its RNA cargo during transportation to the target cells and release the intact RNA into the cytoplasm. So the endosomal escape is a crucial stage to avoid RNA being digested in the lysosomes. However, only less than 2% of siRNA administered in LNPs was previously reported to escape the endosomes and reach the cytosol. So while the importance of endosomal escape for effective intracellular delivery has been established, little is known about the mechanism of interactions between the LNPs and the endosomal membrane. So in the present study, Langmuir monolayers are used as endosomes models to study the interactions with LNPs after the injection into the subphase, with the aim to validate the application of complementary reflectometry techniques to gain new insight into physicochemical processes occurring mainly with respect to pH. In details, so the monitoring surface pressure changes over time gives information on the packing of lipid molecules in the monolayer. Ellipsometry instead exploits the changes of polarized light at the interface, and it is sensitive to the total amount of interfacial material per unit area and its ordering. And finally, Brewster angle microscopy is used to image lateral morphology on the micrometer scale. What we observed, if we look at the results, is that at low pHs, there was a great increase in surface pressure after injection of the nanoparticles underneath the monolayer, mainly indicating lipid insertion into the monolayer. And also, we observe an increase in delta with fluctuations for poly-A-loaded LNPs, mainly indicating the presence of domains. In fact, BAM also showed changes in phase at later time points. And this was not observed with nucleic acid-free LNPs, where only lipid insertion caused the formation of a more homogeneous monolayer. On the other hand, at pHs above 7, much lower surface pressure were reached, indicating a minimal lipid insertion, but some fluctuations in delta were still observed. And we hypothesized that together with the PEG shedding from the coat of the LNPs, the ionizable lipid protonation, which is the main component of the LNPs at low pHs, contributed to LNPs disintegration and the subsequent LNPs component insertion into the monolayer. This does not happen at pHs above 7, where LNPs or their aggregates bind with, to the model membranes. So, reflectivity techniques highlighted for the first time some phenomena occurring in the endosomal environment. These findings prove that these techniques can represent a robust complementary physical basis for future work, comparing a range of newly developed LNP systems with a view to improving the RNA delivery to the cytosol. Thank you, everyone, for listening.
Thank you very much. Before I introduce the next speaker, I would like to ask whether Eduard Gattin is really in the room, because I can't see him. Eduard, if you are here, please tell us. Okay, in that case, Shunpin Han should start getting ready. And now I would like to introduce Aldona Mzuk from UMCG Groningen, the Netherlands. She will talk about nanodiamond magnetometry for real-time monitoring of drug efficiency in arthritis treatment.
Good afternoon.
The floor is yours.
Thank you. So good afternoon, everyone. I hope you can hear me well and you can see my slides in the presenter mode.
Yes.
Okay, perfect. So again, good afternoon. And so thank you for that kind of introduction. So that allows me to move directly to the second slide of mine, where I would like to start with the main goal of this research. So in my project, I will try to detect the free radicals generation in real time in the synovial fluid samples from the arthritis patients using nanodiamond magnetometry. And then later on also prove that this new quantum-based sensing technique could be applied to monitor in real-time efficacy of anti-inflammatory non-steroidal drug like piroxicam. So why we were interested in, or we are interested in the detection of free radicals, because those are active molecules or atoms that possess at least one per electron, and then they are involved, they are crucial for many physiological processes, signaling pathways in our bodies, but at the same time, when they are overproduced, then they cause the oxidative stress in our body that basically leads to or contributes to progression, for example, in osteoarthritis or rheumatoid arthritis. So basically those free radicals at the moment, they haven't been so far used neither as biomarkers nor as the targets for therapeutics. That's because of they are short-lived and very difficult then to detect with the current state-of-the-art methods. But this is like basically why we start developing nanodiamond magnetometry, which is some kind of overcoming this limitation. And this nanodiamond magnetometry is, as I said, quantum-based sensing technique that relies on using the fluorescence, so nanodiamond probes. Those probes possess in the crystalline structure defects, so-called nitrogen vacancy centers, and when they are excited by the green laser pulses of green laser light. They emit red photons, which can be collected so that nanodiamonds can be used as a fluorescence probe. But those optical properties are also linked to very fancy quantum properties. And these quantum properties, in that method, we can polarize electron spins to certain state and then letting them repolarize over time. Once we let them repolarize over time, we can measure time they need for repolarization. We can measure time needed for repolarization. And this is so-called T1 relaxometry. And this is something similar that is measured also in magnetic resonance imaging technique. But in our method, we can do that with a higher nanoscale resolution and using just the confocal microscope. So how it's linked with the detection of free radicals, the method. So we can, once you have a higher concentration of free radicals, you can, near the surrounding the fluorescent nanodiamond, you can detect them, or we see the changes in T1 relaxation time. So higher free radicals concentration, lower T1 values. So we use that phenomena to detect the free radicals concentration in synovial fluid from arthritis patients, but also in the cells in real time, treated also with the diamonds, the cells that were derived from those synovial fluid samples. So what we have found was is that T1 show us that after piroxicam treatments, or in case of osteoarthritis, then we observe the decrease in the free radicals concentration that corresponds to higher T1 values. We haven't observed that phenomena in rheumatoid arthritis samples, so means that those samples or those samples are resistant to the treatment with the piroxicam somehow or not sensitive. We also perform similar research not only for synovial fluid but also cells, as I mentioned. Very similar trends observed for both type of samples after 1 day of the culturing of the cells, but after 7 days we haven't seen any. We are not able to detect any differences between and after treatment with the drug, and we think that's linked with the fact that we just culture cells in a standard medium, so we got rid of all the cytokines and so on that might trigger this free radical generation. To sum up my short talk, our study has shown that fluorescent nanodiamonds can detect free radicals generation in samples from arthritis patients. Magnetometry enables to monitor efficiency of anti-inflammatory drugs, therapeutics in real time, and so what's important that rheumatoid arthritis cells are less responsive to piroxicam treatment than osteoarthritis cells. That's also, I wanted to thank my group, Nanodiamond Group, funding agencies, and you for your kind attention. Thank you.
Thank you very much. Before we continue, Yanira, could you please mute your microphone? Now, Revadi Liamor can start getting ready, but I would like to introduce now Xumping Han from King's College in London, UK. And we will hear about comparative analysis of gold nanorod uptake in mice brain after intranasal administration.
Okay, hello everyone. I hope you can see my slides.
Yes, we do.
Okay, good afternoon everyone. I'm Xumping Han from King's College London. It's a pleasure for me to present part of my PhD work. So the project here is to comprehensively understand the gold nanorod brain uptake following intranasal administration. We know that brain disease treatment is still a challenge due to the presence of the blood-brain barrier. Intranasal administration is an alternative route which can bypass the blood-brain barrier and deliver therapeutic agents to the brain through the olfactory nerve or trigeminal nerve pathway. Gold nanoparticles are attractive materials in nanomedicine. Compared with other morphologies, gold nanorod demonstrates unique optical and biological properties, meaning due to its elongated nanostructure. However, their biodistribution following intranasal administration has never been reported, which is the aim of this project. So in order to track the gold nanorod in vivo, we functionalize the materials either with Cy5, a fluorescent dye, or DTPA, a mental chelator, which can be used for the following radiolabeling study. Here we can see that after functionalization, the particles demonstrate the increased hydrodynamic size, but are still below 100 nanometer and show the decreased data potential compared with the pegylated particles. All the particles had good colloidal stability and showed high road purity. Then we dosed the Cy5 labeled particles in CD1 mice. In the optical imaging, we can see that compared with the control group, all the treated group showed the higher brain signal. However, due to the strong background autofluorescence, it's hard for us to get a quantitative analysis. So then we took the brains and quantified the gold amount by ICP-MS. We confirmed the rapid brain uptake of the particles, and also the gold amount decreased with time increasing. ICP-MS is very accurate. However, it cannot offer the spatial temporal information. To make up for this, we also dosed the radio-labeled particles in CD1 mice and then did the gamma counting as well as the autoradiography study. Here we can see that 10-minute time point shows the highest brain signal, and also the detected signal are mainly located in the olfactory bulb area, and then it shows the deeper transport into other brain regions as a function of time. Also, we noticed a proportion of the particles were swallowed by the animals, and it went through the GI system. So in summary, it is suggested recommended to use a multimodal imaging system to get the quantitative and qualitative information of the gold nanoparticles in biodistribution following intranasal administration, since each model offered the complementary information. So based on this, a solid conclusion can be made. Also, we confirmed the translocation of the particles in brains. So in the future, we will try to conjugate the therapeutic agents onto the particles and do a therapeutic study. Also, the biocompatibility study will be performed to test the safety of the particles. And last, I would like to take this chance to thank my team for all of their support. And also, thank you so much for your listening. Thank you.
Thank you very much.
Yeah, I can stop sharing my screen.
Yes, no problem. Danielle Brain, please start getting ready. Now I would like to introduce another participant from the King's College of London, Revadilia Moore. She will talk about culturing conditions of mesenchymal stem cells, derived exosomes, alter their protein corona formation, cellular uptake, and in vivo organ biodistribution.
Can you see my screen?
Yes, but not in presenter mode yet.
Okay.
Now it's in presenter mode. Thank you.
Okay, good morning, everyone. It is my pleasure to be here to present my PhD work, which is culturing condition of mesenchymal stem cells, derived exosomes, alter their protein corona formation, cellular uptake, and in vivo organ biodistribution. Mesenchymal stem cells, or MSC, has been proposed to be used as an effective therapeutics for liver fibrosis, but the MSC transplantation is limitedly feasible due to low migration and poor cell survival. However, it has been found that the anti-inflammatory properties and anti-fibrotic effects can be mediated through paracrine effects of exosomes, 100 nanometers extracellular vesicle. Exosomes like liposomes, they can be modified to enhance targeting potential. Also, the dose and quality can be optimized and more easily controlled. The aim of my PhD work is to characterize MSC-derived exosomes, MSC-derived exosome or MSC-exo from different culturing methods and study their impacts on pharmacokinetics and dynamics. We hypothesize that the targeting and therapeutic properties of MSC-exo can be enhanced by optimization of culturing conditions to obtain favorable biodistribution and cellular targeting. The MSC-exo derived from method 1 and method 2, they are identical in size, as can be seen from the nanoparticle tracking analysis result. And also they are identical in the surface chart and the expression of exosomal markers, which are CD81, CD9, CD63, and TSG101. By contrast, The difference in culturing conditions seem to affect the protein identity and the protein corona formation, as can be seen from the SDS page result. Also, the difference in vitro cellular uptake and the mediated anti-inflammatory activity can be observed, that i.e. the MSC-XO2 seem to possess superior anti-inflammatory properties evaluated in human-derived macrophages when compared to MSC-XO1. Interestingly, when looking deeper into in vivo uptake by subpopulation in the liver, we can find that the MSC-XO2 can be taken up more by hepatocytes, endothelial cells, and stellar cells, whereas no difference in the uptake by Kupffer cells can be observed here. For my future work, I will be focusing on the correlation between protein corona composition and cellular targeting. Also, the therapeutic efficiency mediated by both exosome types will be evaluated in the animal models with liver fibrosis induced by carbon tetrachloride. The findings from my PhD work might offer an alternative way to modify exosomes for targeting purpose by optimizing culturing conditions to promote favorable protein absorption that can enhance cellular targeting and prevent progression of diseases. I would like to thank my supervisor, Professor Khulud Al-Zaman, and my colleagues, and thank you everyone for listening.
Thank you very much. Marijke Duyker, you will be the one after. Now I would like to introduce Danielle Brain from the University of Liverpool in the UK, and she will talk about assessment of cell phenotype following repeat exposure to NRTIS, FTC, and 3TC.
Thank you for your kind introduction. Here I'm just going to briefly introduce my poster looking at the assessment of cell phenotype following repeat exposure to the nucleoside reverse transcriptase inhibitors FDC and 3TC. If you'd like to find out some more information, you can check out my poster in the Clinam Poster Lounge, and feel free to ask me any questions later. I'm a final year PhD student and research associate in the immunocompatibility group at the University of Liverpool, and our group focuses on the immune interactions and safety of advanced therapeutics such as nanomedicines. My group has been involved in a number of nanomedicine consortiums such as EU-NCL, Refine, and Safer MedTech, and we are also part of the Centre of Excellence for Long-Acting Therapeutics, CALT. One project under CALT that I've been involved with during my PhD has been to develop and assess the safety of novel long-acting drug-release therapies for the treatment of HIV. Biocompatibility assessment of these materials is particularly important due to the previous issues with biodegradable TAF implants to treat HIV. So for this work, I repeatedly exposed 4 cell lines, THP1s, MUTS3, CEM, and KU812s to FDC and 3TC at the Cmax concentrations for 7 weeks with passages twice weekly. I then carried out cell health assessments, measuring ROS using the CellRock screen reagent, measuring reduced glutathione using the PhioTracker Violet reagent, and mitochondrial membrane potential using the JC1 reagent. And then after staining and washing, these were all analysed using flow cytometry. I then also exposed each of the repeatedly exposed cells from each cell line to known positive activation controls. For THP1s, this was LPS. MUTS3, we use LPS or resiquimod. For KU812s, the E5A or calcium isonophore, and for CEMs, I used PHA. And then following the exposure, I then stained and assessed the expression of cell-specific activation markers using flow cytometry. I found that the impact on cell health, as well as marker expression following exposure to positive activation controls, was cell line specific. So one example from this work where I used the THP1 monocytic cell line, I found that repeat exposure to 3TC resulted in oxidative stress as evidenced by greater ROS, greater reduced glutathione, mitochondrial membrane depolarization, and also greater CD14 expression. This was both when the 3TC repeat exposed cells were untreated, and also when they were, it was also increased when they were exposed to LPS as well. So I found that repeat exposure to the antiretrovirals FTC and 3TC alters cell phenotype and has immunomodulatory consequences following exposure to subsequent immunological challenges. Therefore, consideration of the active pharmaceutical ingredient is therefore necessary on top of testing the carrier for biocompatibility testing for long-acting implantables and also injectables for future medical use. Thank you for listening.
Thank you very much. Bettina Heiss, you can start getting prepared. And now I would like to introduce Mareike Deuker from the Max Planck Institute for Polymer Research in Germany. And she will talk about interaction of nanocarriers with anti-PEG antibodies. The floor is yours. Maybe you are muted. We can't hear you.
Thank you very much. Can you hear me now?
Yeah. Yes. Yes.
Perfect. Thank you. So yeah, thank you very much for the nice introduction. My name is Mareike Deuker. I'm currently a PhD candidate at the Max Planck Institute for Polymer Research, and my short presentation is about the interaction of nanocarriers with anti-PEG antibodies. Polyethylene glycol is still the gold standard for many biomedical applications. It is very famous and widely used for its stealth effect. Through pegylation of nanocarriers, their pharmacokinetics and dynamics can be improved. For example, the increased water solubility, decreased enzymatic degradation, and overall prolonged circulation time. Nevertheless, there are increasing reports that PEG-binding antibodies can be elicited in humans. In contrast to most anti-drug antibodies, anti-PEG antibodies were observed in both pegylated therapeutics and treatment-naive individuals. Various research groups observed that the administration of repeated doses of pegylated nanocarriers led to an accelerated blood clearance and weakened the efficiency of pegylated therapeutics. Additionally, hypersensitivity reactions to PEG and acute severe allergic reactions have been observed. So despite the potential serious consequences of circulating anti-PEG antibodies, their influence on the effects of therapeutics and on related side effects remains an unanswered question. To address this question, a detailed study of pre-existing anti-PEG antibodies in healthy individuals among the German population was performed using an ELISA. We received 500 blood samples from the University Clinic Mainz and analyzed them on the anti-PEG IgG and IgM concentration. Here you can see representative sum results for the anti-PEG IgG analysis. A high prevalence and concentration of anti-PEG IgG and IgM was found throughout all the samples. Notably, the concentration and prevalence decreased with increasing age. Overall, anti-PEG antibodies could be found in over 62% of the population, where over 80% were actually positive for anti-PEG IgG and a little bit less were positive for anti-PEG IgM. To further evaluate the biological response, the role of anti-PEG antibodies in the protein corona was investigated. The protein corona is the biological coding of the nanocarrier that created its biological identity as recognized by the cells. We investigated the enrichment of anti-PEG antibodies in the protein corona of pegylated silica nanocapsules. Therefore, the silica nanocapsules with PEG in different chain lengths and surface densities were prepared and to analyze the influence on the protein corona formation. An enrichment of anti-PEG antibodies in the protein corona of pegylated nanocarriers compared to pegylated nanocarriers to non-pegylated nanocarriers could be observed. Notably, also a minimum chain length is necessary for anti-PEG antibody binding with a threshold of a certain length, then there is no increase in anti-PEG antibody binding anymore. Additionally, the cellular uptake of pegylated nanocarriers with varying amounts of bound anti-PEG antibodies will be monitored. Here you can see some preliminary results. We prepared silica nanocapsules with different amount of anti-PEG IgG in the protein corona and subsequently analyzed the cell uptake in macrophages. You can see that the cell uptake in macrophages increased with the anti-PEG antibody concentration in the protein corona. These results suggest that the existence and concentration of anti-PEG antibodies in the protein corona of different pegylated and non-pegylated nanocarriers should be further evaluated to determine the potential effects also in vivo. And with this, I would like to thank you all for your attention, and I'm happy to discuss any questions later on.
Thank you very much. Now, since Valentina Franchia had to leave the session, I would ask Manuela Gaspar to be prepared. Now I would like to introduce Bettina Heiss from the Hospital of University of Munich in Germany. She will talk about enhancing drug efficacy with a heat-activated drug delivery platform based on phosphatidyl oligoglycerol nanocarrier.
All right.
The floor is yours.
Thank you. Thanks for the nice introduction. I'm a PhD student at the LMU and the working group Agiliposum of Professor Lars Lindner. And as you just mentioned, I will be focusing on our heat-activated drug delivery platform. So first of all, to get everyone on the same page, I would like to say that our lipid nanocarriers are based on 3 lipids, which is the SPC, DPPC, and DPPGN. And we are able to form lipid bilayers with certain characteristics as a certain shape as well as size and a specific melting temperature of the lipids, which enables an ultra-fast heat-triggered drug release at temperatures above 40 degrees. This was not only shown in vitro, but also in vivo, where we were able to see a sustained plasma stability of our liposomal formulation, but an efficient ultra-fast heat-triggered drug release at temperatures of the mild hypothermia range, which would be 41 to 43 degrees. And this is also the proof that our lipid nanocarrier is a perfect fit to combine with this mild hypothermia. The advantage of hypothermia is not only that it's giving us a release mechanism, but also that it's already in clinical translation for a combination of therapy with standard of care chemotherapeutics in example, sarcoma treatments. And another advantage of hypothermia is that it's increasing the perfusion of the heated area as well as the drug uptake into the heated cells. This was shown also from us in a rodent tumor model, and here we were able to have a 15-fold increased DOX concentration in heated tumor compared to unheated tumor tissue, and especially in comparison to clinically relevant controls, which would be free DOX, thermodox mimicking formulation, or Helix. We were able to show that with our formulation, the drug content was significantly increased in the heated tumor tissue, which then this also nicely translated into a prolongation of survival. In contrast to the 5 milligram per kilogram that were IV given for Helix and Freedox, our formulation was given with 2 milligram per kilogram, and still the therapeutic efficacy was significant. And this data was giving hope. So a 2-arm comparative oncology trial in cats was conducted. Here the felines were bearing a spontaneous feline fibrosarcoma, and the 2 arms were made of our TSL dox formulation and a free dox group. Both drugs were given IV via infusion, and for the first treatment cycle, safety was assessed, and the pharmacokinetic profile that revealed a sustained plasma stability of the liposomal formulation also in the cat's patient. Then furthermore, the drug infusion was combined with local hypothermia application, which was given via an RF antenna. This was done 6 times, and final a surgery was planned to resect the tumor, and subsequent PET MRIs were taken to follow the tumor growth. Overall, we can say that 3 out of 4 animals of both groups were able to conclude the 6th cycle. Unfortunately, 1 animal of the 3 dogs group developed lung metastasis, which is why tumor resection was not conducted. And all of the animals of our TSL dogs group underwent surgery in the end, and the tumors that were resected show 10 to 20% residual vital tumor cells, which is in line with the PET MRI scans where all of the tumors showed metabolic partial response, which means that all the tumors went into regression, in contrast to our 3 dogs groups where the tumors rather went into progression. So overall, the seen effects in the cats was very promising, and further clinical translation is planned for the end of the year. And also, of course, our group is focusing on encapsulating new drugs in order to develop nanocarriers for further preclinical testing. And to sum up my speech, I would like to thank the jury of the poster prize, as my poster was awarded with the first prize in the topic of translational nanomedicine priorities. And I'd like to thank you for your attention.
Thank you very much, and congratulations for the prize.
Thank you.
Satinder Deb Kaur, you can start getting ready. Now I would like to introduce Manuela Gaspar from the University of Lisboa in Portugal. And she will talk about liposomes, a highly efficient drug delivery system for different clinical applications.
Thank you.
Your slide is not in presentation mode.
It's not? And
no.
Now? It is.
Yes, now it is, yes. Thank you.
Thank you. So I'm a good morning, everybody. I'm a researcher from the Research Institute for Medicines from the University of Lisbon. And I belong I'm included in the Advanced Technologies for Drug Delivery group. And our research group, the major challenge is to deliver bioactive entities at cellular and intracellular target sites. And actually to improve this delivery, not only in vitro, but also in vivo, the association of potential bioactive entities to carrier systems has been used. And among the drug delivery systems, our preferential are liposomes. And using liposomes, this strategy has been successfully developed to achieve an efficient macrophage-mediated drug delivery in case of mycobacterial or parasitic diseases, or to reach tumor sites, as an example in melanoma. Here, I'm showing an example of an antibiotic, rifabutin, that was incorporated in liposomes. And what we observed was that in a Mycobacterium avio model of infection, to target rifabutin to liver and spleen, rifabutin liposomes prepared with fluid phospholipids and with a mean size of around 300 nanometers was able to reduce the bacterial loads in a much higher extent than free rifabutin. In another murine model, the Mycobacterium tuberculosis model, we changed the lipid composition and we used rigid phospholipids and with a lower mean size and we were also able to have very good effects in comparison with the antibiotic in the free form. In another example, deals with the use of paromomycin that was incorporated in liposomes and tested in a murine model of leishmaniasis. And what we observed was when this antibiotic was incorporated in liposomes, a huge reduction in parasite burden was observed for animals treated with paramomycin liposomes in liver and in spleen, while when the drug was administered in the free form and using the highest dose, very low, modest results were observed. In addition, we have compared paramomycin liposomes versus ambisome, the liposomal formulation of amphotericin that is approved in clinical for treatment of leishmania infections, and similar parasite burden reduction between paramomycin and ambisome were achieved, of course, using different doses. In another example, where I would like to show when a copper-based complex was incorporated in liposomes and tested in a melanoma model. And the liposomal formulation of this copper-based complex exhibit the highest reduction tumor progression as a result of liposomal accumulation at tumor sites. So, in conclusions, where I would say that liposomes of rifabutin and paramomycin resulted in improved accumulation of loaded antibiotics at infection sites and consequently enhanced therapeutic effects, that paramomycin liposomes may be an alternative to ambisome, and the use of long blood circulating liposomes for loading a copper-based complex were able to extravasate at tumor sites and reduce tumor progression. Of course, for each formulation, we have to select the most appropriate lipid composition for liposomes preparation. And thank you for your attention.
Thank you very much. Soraya Pinto, you can start getting ready. Now I would like to introduce Satinder Deb Kaur from the Nottingham Trent University in the UK. And she will talk about mechanism behind selective infiltration of lipid nanoparticles in the spleen following ischemic stroke. Please, the floor is yours.
Hi, thank you very much for the kind introduction. I'm a PhD candidate at Nottingham Trent University. So today I will talk about mechanism behind selective infiltration of liposomes in the spleen after ischemic stroke. We are interested to understand the splenic response to acute brain injury. While over the years, we got to understand that brain and spleen has a bidirectional communication after stroke. To highlight, after stroke, HP axis and sympathetic nervous system gets activated, which cause release of catecholamines like norepinephrine. So spleen gets directly innervated with norepinephrines, which cause shrinkage of spleen and cause apoptotic loss of splenocytes like marginal zone B cells, which are our main cells of interest. So loss of these marginal zone B cell cause loss of circulating IgM antibodies and impaired antibacterial defense mechanism of the host, which impaired and which make the patient susceptible to post-stroke infections, particularly pneumonia, which has a higher mortality rate after stroke. On the other hand, shrinkage of spleen causes efflux of splenocytes and cytokines into the systemic circulation, and from where it migrate into the brain at different time intervals, and that's how it causes neuronal damage and disrupt blood-brain barrier. Therefore, ability to manipulate these peripheral immune changes in the spleen without inducing off-target effect could represent an attractive treatment strategy for many neurological disorders along with stroke. I recently published my review, so if you are interested to understand more about the area and possible nanomedicine approaches, please go and visit. Recently, we have seen selective distribution of lipid nanoparticles in the spleen after stroke in a mice underwent middle cerebral artery occlusion. So we have seen distribution is significant in 24-hour and 48 hours time span window. Although we have seen liposomes are going into the spleen and earlier time points of MCO as well. And another interesting fact is we have seen this distribution of liposome is more selective to white pulp and the marginal zone area of the spleen. So further, we wanted to investigate why and the reason behind this selective liposome accumulation in the spleen after stroke. So we started to look at different parameters. So we have seen changes in splenic structure after stroke and enhancement in blood vessel integrity. Also, we have seen that phagocytosis capacity of splenic macrophages enhanced, which could be another reason for accessibility of nanoparticles to the spleen after stroke. And another interesting fact, we have seen ACM degradation of spleen after stroke, which is quite new in the area. I have given more detail about ECM changes and ECM remodeling action after stroke in my poster. Please go and visit my poster in poster launch. So for future translation, we know that liposomes are going into the spleen, possibly because of all these parameters. So we will take advantage of it and we will encapsulate caspase inhibitors in the liposomes to prevent apoptotic loss of splenic marginal zone B cells to prevent post-stroke infections. At the end, I would like to thank team and people involved in this work. Also, I would like to thank organizers to award my poster first prize under basic basic nanomedicine priorities. I'm happy to take questions at the end. Thank you very much.
Thank you very much, and congratulations to you too for the prize. Elena Garde, you can start getting prepared. Now I would like to introduce Soraya Pinto from I3S at the University of Porto in Portugal. She will talk about FcRn targeted nanomedicines for intestinal delivery of an anti-diabetic peptide.
Thank you, Ruth, for the nice presentation. Good afternoon, everyone. My name is Soraya Pinto. I'm a PhD student at I3S, University of Porto in Portugal, and my work is entitled FcRn Target Nanomedicines for Intestinal Delivery of an Anti-Diabetic Peptide. So the main goal of my project is to improve the oral bioavailability of anti-diabetic peptide. And for that, I will encapsulate the anti-diabetic peptide inside of polymeric nanoparticles. And after that, I will functionalize surface of the nanoparticles with ligands that target a receptor located in the membrane surface of the enterocytes, the FcRn. The binding between the nanoparticles and FcRn occurs in a pH-dependent manner, which means that the binding between the nanoparticles and the FcRn occurs at acidic pH, which is found in intestinal lumen, and this connection between the nanoparticles and FcRn occur when exposed at neutral pH, which is found in the lamina propria. So basically, the mechanism is just like that. So after the nanoparticles achieve the intestinal lumen, the acidic pH will favor the binding between the nanoparticles and FcRn. After that, the complex will be internalized by endocytosis for the enterocytes, and then the acidic endosome of the acidic endosome will be responsible to favor the maintenance of the binding between the nanoparticles as well as the FcRn. The endosome will be transported through the enterocyte by transcytosis, and after that will fuse with the other side of the membrane of the enterocytes, and the interaction with the physiological pH of the lamina propria will be responsible to promote the release of the nanoparticles from the FcRn. So this mechanism is principally responsible to protect nanoparticles from the lysosomal catabolism, and in this way promote the intestinal transcytosis of these nanosystems. So to achieve the main goal of this project, we select FcRn target ligands from the literature. We evaluate in-house their binding to the human FcRn by performing SPR, and the outcomes demonstrate that only these two FcRn target ligands, this peptide FCBP3 and this protein, were responsible to bind to the human FcRn in a pH-dependent manner, so bind at pH 6 and non-binding was detected at pH 7.4. After that, we functionalize the PLJ pegmolamide, which is a copolymer that we use to produce the PLJ nanoparticles, and we functionalize this polymer with both FcRn target ligands, the Affibody and the peptide, and evaluate the conjugation efficiency of them. Regarding the conjugation efficiency that we obtained for the peptide was around 79%, and for the Affibody only 16% was obtained. After that, we produce non-functionalized nanoparticles by double emission technique and physical-chemical characterize these nanoparticles in terms of average size, PDI, zeta potential, association efficiency, and loading of the antidiabetic peptides. Regarding the association efficiency and drug loading of the antidiabetic peptide, the values obtained were around 78% and 4% respectively. As future perspective, we will produce functionalized nanoparticles with both FcRn target ligands. After that, we will evaluate the binding affinity of those functionalized nanoparticles to the human FcRn by performing SPR and in vitro uptake studies, including flow cytometry and confocal microscopy. And after that, we will evaluate the intestinal permeability of those functionalized nanoparticles and compare with non-functionalized nanoparticles by using a 3D intestinal model that was developed in our group. So I would like to thank all of you for your attention. I would like to thank all my team members, Precision Nanomedicine for the fellowship to participate in this conference, Novo Nordisk for kindly supply us the antidiabetic peptide, and FCT for my PhD scholarship. So thank you all.
Thank you very much. Emili Laprevot, you can start getting ready. Now I would like to introduce Elena Garday from Jena University Hospital in Germany, and she will talk about selective uptake into inflamed human intestinal tissue and immune cell targeting by worm-like polymer mice cells.
Thank you for a kind introduction. I would like to show you our project where we're focusing on targeted nanoparticles for efficient translocation across gastrointestinal barrier. Current treatment of patients with inflammatory bowel disease unfortunately cannot sufficiently reduce all symptoms and very often induce drug-related side effects. But using nanoparticles, we can deliver drug only in inflamed area of intestine without interactions with healthy tissue. In our project, we use one polymer and formulated 3 different morphologies: very small spherical nanoparticles or micelles, very large vesicles, and worm-like nanoparticles or phylomicelles. And we investigated how these 3 different morphologies influence and which impact it has on penetration across human mucosa. For this aim, we used biopsies from patients with inflammatory bowel disease or from control healthy donors, and we mounted the biopsies in ozone chambers and apply equal amount of nanoparticles in luminal part of ozone chambers. So, small spherical nanoparticles demonstrated very fast uptake by healthy and inflamed tissue without significant difference between 2 groups. In contrast, large vesicles didn't demonstrate sufficient uptake by human mucosa. And noticeable difference we observed when phylomicelles were applied, demonstrated their selectivity for inflamed tissue. After experiment, we cut the biopsies and investigated the localization of nanoparticles in healthy human mucosa and in inflamed. So we stain epithelial cells with ecadherine, we stain all nuclei with DAPI in blue. Our particles are in green color, and in lamina propria immune cells we stain with CD11B in red color.
As you can see, spherical nanoparticles target immune cells in lamina propria in both healthy and inflamed tissue. In contrast, again, phyllomicelles, they were observed only in inflamed tissue, also with targeting immune cells. And it's important to notice that actually selectivity and targeting immune cells, especially macrophages, is very crucial for further treatment of patients with IBD. Thank you so much for your attention.
Thank you very much. Rafael Baker, you can start getting ready. Now I would like to introduce Emili Laprevot from Perseo Pharma AG in Switzerland. And she will talk about gut homing stable enzyme activities to develop efficient digestive therapies.
Thank you, and good afternoon, everybody. I'm Emili Laprevot, the CDO of Perseo Pharma. So at Perseo Pharma, we develop metabolic therapies, and today we're going to make a focus on one of the applications for digestive therapies. So, does it move? Yeah. So enzymes are the pillar of digestion, meaning that they are responsible of the quality of our nutrition. So for patients with faulty digestions, enzymes have been developed for therapeutic purposes, but the existing formulation of enzymes that are available today do not satisfy the patient because they exhibit a poor drug profile. And that is due to the poor stability of the enzyme, a high susceptibility of the enzyme to the harsh condition in the gastrointestinal tract, a lack of specific localization in the intestine, and a variable intolerance profile that they trigger because of the huge amount of enzymes that the patient has to take to compensate the low activity of the enzymes. So there's here a real unmet medical need for this patient with digestive disorders. So at Perseo Pharma, we developed a therapeutic enzyme platform to coat the intestine with a sustained enzymatic activity. And so it consists in a nanomedicine made of a core silica nanoparticles on which enzymes are covalently bound, so there's no release mechanisms. And these enzymes are shielded with an organosilane layer. So this layer is soft and porous, so the enzyme remains fully active within the layer, and the substrate can migrate through the layer to reach the active site of the enzymes. And additionally, this layer allows to isolate the enzyme from the environment, so they are not exposed to any external stresses. And additionally, in order to induce a specific localization of the nanomedicine, we functionalize the surface of the nanoparticles. So today, with the example of today, with mucoadhesive moieties, in order to induce an interaction between the nanomedicine and the mucus that covers the wall of the intestine. And so here we report some preclinical results showing that the nanomedicine exhibits a sustained enzymatic activity in presence of proteases. So proteases to mimic the harsh condition in the gut environment. And when compared to the free enzyme, we see that the enzyme on the nanomedicine is not susceptible to proteolytic degradations. In terms of biocompatibility, we demonstrated the safety of the use of the nanomedicine for gastrointestinal application in vitro, so that you can see all the results in the poster. And we confirmed it in vivo on mice. So after oral administration of the nanomedicine on mice, the particles remained in the gastrointestinal tract and they do not become systemic. That is really important. And after 24 hours, all the nanomedicine are excreted. That is in accordance with the turnover of the mucus in mice. Finally, we demonstrated the importance of the functionalization of the surface of the nanoparticles for its interaction with the mucus. So you can see here on the SPECT-CT images from Göttingen mini pigs dosed with the nanoparticles that we have a localized control localization of the nanoparticles within the intestine. And when we compare with the non-functionalized nanoparticles, we can notice that we increase the residence time of the functionalized nanoparticles in the small intestine from a factor 3. And I remember you that the intestine is the site of action of the nanoparticles and the enzymatic activity. So here we show that we have nanomedicine that can coat the wall of the intestine with a sustained enzymatic activity. And we are now engaged in the development of a series of novel drug compounds, and some of which will be co-developed together with Nestlé Health. Thank you for your attention.
Thank you very much. Before I introduce the next speaker, I would like to ask whether Hinasom Akbu is in the session. It doesn't seem like that. That means that Katharina, you can start getting ready again. And now I would like to introduce Rafael Baker from King's College London in the UK. And she will talk about nose-to-brain delivery of riluzole-loaded polymeric nanoparticles for the treatment of amyotrophic lateral sclerosis. The floor is yours.
Hello, and thank you for taking the time to listen to my talk today. As mentioned, my name is Rafael, and my project focuses on the development of polymeric nanoparticles for the treatment of amyotrophic lateral sclerosis, also known as ALS. ALS is a progressive heterogeneous disorder with a complicated etiology where about 10% is genetic and the remaining 90% is sporadic. And the hallmark of ALS is the degeneration that is seen in both upper and lower motor neurons and frontal cortex neurons in varying degrees. Therefore, this results in a spectrum of ALS to frontotemporal dementia, FTD, thus presentations of the disease are individualized based on the involvement of each element. And sadly, this fatal disorder is still incurable. The current standard treatment regimen is a multidisciplinary approach to slow down disease progression, which involves the use of oral riluzole, a glutamate inhibitor, alongside other interventions shown here. And this approach has its challenges, mainly due to riluzole's poor water solubility, poor efficacy, and problems in patient adherence. We therefore hypothesize that by combining the benefits of the nose-to-brain route, which is a convenient, non-invasive direct route to the CNS, mainly through the olfactory and trigeminal nerves, bypassing the BBB, blood-brain barrier, and combining that with improvements in drug solubility and the pharmacokinetic profile that is offered by a nanoparticulate system, we can improve CNS uptake and potentially enhance therapy for ALS patients. We started formulating riluzole PLGA nanoparticles using nanoprecipitation methods, and we have achieved appropriate characteristics for intranasal administration. TEM images shown here confirm that our riluzole nanoparticles are monodispersed, spherical in shape, and the entrapment efficiency we measured was about 20%, which can be used to dose animals in vivo. Then, radiolabeling of the drug was sought for in vivo quantification, which was confirmed using multiple techniques shown here, and the specific radioactivity was sufficient for in vivo quantification using liquid scintillation counting. Riluzole biodistribution in vivo in major organs by different routes was evaluated, and when we took a closer look, we found that intranasal riluzole nanoparticles achieved therapeutic concentration in the brain within 10 to 30 minutes, similar to that of the invasive intraperitoneal route, which was significantly higher than oral delivery. And we found that systemic absorption of riluzole post-intranasal delivery was unavoidable due to ingestion, but it can enable an additional access route to the brain via the BBB. The next step is to evaluate the utility of intranasal riluzole nanoparticles for ALS, and ongoing therapy studies will conclude on that. In summary, we showed that our approach has potential for clinical application. By utilizing this convenient route, we can efficiently target the brain, bypassing the first pass metabolism, and by our biocompatible formulation can be used as a vehicle for a variety of drugs. And finally, I would like to acknowledge my group and our collaborators. And thank you for your attention.
Thank you very much. So now I would like to introduce once more Katarina Spiridopoulou from Democritus University of Thrace in Greece. talking about diverse bioactivities of biogenic selenium nanoparticles spur cancer cell-based vaccine potential. And now it looks perfect.
Hello, everyone. Just to be sure, can you see my slides change?
Yes. At least it changed from, it changed once. Try to change once more, just to see. No, nothing is happening.
Hello?
Hello?
Can you hear me? I don't seem to hear you.
I can hear you now. I think you were, yeah, I think you were frozen. Yeah, now it works.
Okay.
I think it works
okay.
now.
So the slides change, right?
Yes, yes.
Great, great. Okay, let's go now that we have it. So hello, everyone, once again. I'm really sorry for the technical problem. So what I've been trying to talk to you about today is about the diverse bioactivities of biogenic selenium nanoparticles that spur cancer cell-based vaccine potential. So why selenium? Selenium exerts multiple health-promoting effects. Among those are immunomodulatory effects and anti-cancer effects, but unfortunately, it's toxic in higher doses. So selenium nanoparticles, it's a more bioavailable and more biosafe alternative form of selenium. So we achieved biogenic synthesis of selenium nanoparticles by utilizing the probiotic strain Lactobacillus casei 80cc393. And why go with the biogenic synthesis? Because first, safety, and second, the variety and the specificity of microbial enzymes used in biosynthesis cannot be achieved by chemical synthesis approaches. So we chose this specific strain because we were based on our previous work, and we have seen that it itself exerts anti-tumor effects against colon cancer, and it can accumulate selenium. So the outline of our work, we used the probiotic strain in order to synthesize nanoparticles, extract it and isolate it and characterize them. And we investigated their anti-cancer effects in cancer cells. And also we employed the syngeneic CT26 colon cancer model in biopsy mice in order to evaluate the anti-tumor potential of the nanoparticles in vivo. So what did we see? The nanoparticles we extracted were spherical with an average of 360 nanometers in diameter, and they were associated with bacteria-derived biomolecules. We assayed their bioactivities in colon cancer cell lines, in human biopsies, and in murine primary cells. And we report that the nanoparticles exert cancer-specific antiproliferative effects. They induced apoptosis in cancer cells, evident by K-space 3 activation and by the modulation of protein levels of molecules involved in apoptosis, such as the BCL2 family members. And more importantly, the nanoparticles were found to induce immunogenic cell death in cancer cells. All the main hallmarks of immunogenic cell death were observed in treated cells, namely the release of HSGB1 and ATP, the translocation of calreticulin and ERP57, and also the secretion of pro-inflammatory cytokines from treated cells. So these interesting results led us to our in vivo experiments. We used the same syngeneic tumor model in biopsy mice in two different experimental settings. First, we orally administered the nanoparticle in mice prior tumor challenge, and we observed a 50% tumor volume inhibition. And second, in the second experimental setting, mice received vaccines that were prepared, that were comprised of
tumor cells that had been pre-treated with the nanoparticles. And mice received those vaccines prior tumor challenge. And as you may see, tumor growth rate of the animals in the group that received the vaccine was confined compared to the control animals. So our results indicate that these biogenic nanoparticles could pose the basis for the development of a cancer vaccine with preventive and even therapeutic efficacy. These vaccines, these whole cell cancer vaccines, comprised of tumor cells treated in order to be inactivated or for their immunogenicity in order to be enhanced, were among the first vaccines investigated against cancer, but did not perform well in clinical studies. So there is a seek in trying to identify new immunogenic cell death markers. And our results highlight that these biogenic nanoparticles hold a great promise as such immunogenic cell death agents. Thank you for your attention. I'll be more than happy to take questions later, and enjoy the conference and see you in the next sessions.
Thank you very much. Now, since 3 presenters didn't show up, we are in fact, we managed to finish in time after this long marathon. What I would like to do is I would really like to thank everybody for clear and good and interesting presentations, and almost all of you managed the time, which I really like. I hope that you all come now to the debate lunch too. There is a link in the chat, so you can just click on the link and then move to this debate lunch, and there we can interact and ask questions and discuss. So thank you very much. And with this, I would like to conclude the session 5, the small speech session.